Hello everyone, welcome to Mastermind. In this video, let us discuss about biotechnology and this is part 3. So in part 2, we have already discussed about the conceptual development of principles of genetic engineering. So after that, in this part, we'll be discussing about the recombinant DNA technology. So this video especially will be very interesting for you to understand, and in this video, we'll discuss about restriction enzyme. This will be important part of our general studies in paper 3 under the biotechnology section, and also it will be helpful for us in answering prelims related questions on general science, and if the question is related to biotechnology, then it will be easier for us to answer as well. Now, in order to continue from the principles of genetic engineering, there we saw about molecular seizures, or we called it as restriction enzymes. We saw what is the cloning, etc. Now, in this recombinant DNA technology, recombinant, that is recombining DNA technology, there are different tools which are used to achieve this recombination. Okay, there are different tools which are used in order to achieve this recombination. So some of the key tools are, first one is restriction enzymes. First one is restriction enzymes. The second would be polymerase enzymes. Third one would be ligases. Fourth one is vectors. And last one is host organism. So in this particular video, we'll be discussing about restriction enzymes. Now, before we process into understanding restriction enzyme, which is very easy for us to understand, there is small basics with respect to DNA. Let us just glance across that. So whenever we are considering DNA, so let us say this is an example for DNA. Okay, this is an example for DNA. So whenever we are considering DNA as a molecule, there are certain bases which are found in a DNA molecule. There are certain bases which are found, that is, they attach with each other, and you can call it as ACGT. Now what is this ACGT? In ACGT, A stands for adenine. C stands for cytosine. G stands for guanine. And T stands for thymine. Okay, whenever we are looking at any DNA sequence, on that there are lot of bases which are found, and they are only 4, that is adenine, cytosine, guanine and thymine. For now, to understand, only these will be there. Now any DNA if you consider, then there are two strands which are going. Okay, here this is one strand and this one is another strand. Now let us take this and wind it up like this. Just an example for us to understand more. Now you have to always remember that there are pairs. A always pairs with T, and C or G always pairs with C. Similarly, T always pairs with A, C always pairs with G. That is, adenine always pairs with thymine, cytosine pairs with guanine. Now let us take one example. Here, let us consider this to be one strand. Now let us take A, C, T, G, C. In the opposite, if A is there, then you will have T. C is there, you will have G. Again, C is there, you will have G. T is there, you will have A. G is there, you have C. G is there, you have C. And C is there, you have G. So A always pairs with T. C always pairs with G. So the sequence will be changing. So this is the basics for us to understand. What is this? This is ACGT, which is the acronym for 4 types of bases which are found in DNA molecule. So I hope this part is very clear. Now let us see what are restriction enzyme. Like we study science, we have to divide all the words into individual words. Now there is some restriction and there is enzyme. That is why it is called as restriction enzyme. In order to understand this, let us take an example for bacteria and understand how a virus attacks bacteria. Okay, how a virus attacks bacteria. Now let us take one bacteria. So let us take an example for bacteria. Now this bacteria has its own DNA. Correct? This is bacteria, and the bacteria has its own DNA. Let us call this as bacterial DNA. Let us call this as bacterial DNA. Now outside there is a virus. Now the virus found out that the bacteria is present. So virus, what it does is it injects the bacteria with its own DNA. With its own DNA. Now there are two DNAs in bacteria. One is the bacterial DNA and other one is the DNA which has come from virus. That is, virus has attacked. Virus has attacked the bacteria. So this is viral DNA. Okay, so let us say this is virus DNA. Okay, now there are two DNAs. One is bacteria DNA and the other one is virus DNA. Now the process starts. First thing what the bacteria has to do is to destroy virus DNA. Correct? Bacteria must destroy what? The virus DNA. The virus DNA. So the first process what bacteria will do is label its own DNA. So bacteria will label its own DNA. Its own DNA, so that it is easy for it to recognize. That is, this is my DNA. Example, this is my DNA. It will say, and it will label its own DNA. Now this labeling is done by synthesizing methylase. This is done by synthesizing methylase. Okay, now when the DNA, bacterial DNA, now when bacterial DNA synthesizes methylase, then there will be labeling of this particular DNA. Okay, then there will be labeling of this particular DNA. Now because it is labeled, what are these called? These labels, these are called as methyl groups. These labels are called as methyl groups. So the first process what bacteria did, it wanted to label its own DNA. How did it label its own DNA? By DNA synthesis of methylase. Now it will have methyl groups around its own DNA. Now because it is having methyl groups around its own DNA, it will be able to identify which is the bacterial DNA and which is the viral DNA. Now how will it identify which is viral DNA? The DNA which is not having methyl groups. The DNA without methyl groups. Without methyl group is the viral DNA. Correct? That is the difference from which it is coming. I hope till here it is very clear. So any DNA which doesn't have this methyl groups attached is identified as the viral DNA. Now there is something called as restriction enzyme. This restriction enzyme identifies within this bacteria that identifies the bacterial DNA and it identifies the viral DNA. So it identifies bacterial DNA and also identifies viral DNA. So it knows that viral DNA does not belong over here. And hence this restriction enzyme destroys this viral DNA, and this is how the bacteria protects itself from the viruses. So the restriction enzyme restricts the growth of viruses. Now, in order to understand this, why did we discuss about the basics of this DNA? That is the bases found which are in the DNA molecule. That is because this restriction enzyme restricts the growth of viruses. Now, in order to recognize or identify, it needs specific sequence of bacteria. Now, let us take bacteria's sequence. Let us take the sequence of bacteria. The sequence in the sense the ones which we discussed before. Now let us say the bacteria has this sequence which is CT, AT, AG. This is just an example. Okay. Now in place of C, opposite to C, we need G. In opposite to T, we need A. Again T, A, T, C. Now if you can read it from the top, C, T, A, T, A, G. Bottom, C, T, A, T, A, G. So this is called as palindrome. This is called as palindrome sequence. This is called as palindrome sequence. Now what is palindrome? For example, 1661. This is palindrome because if I read it in this manner or from this side, it is 1661. From this side also it is 1661. So it is called as palindrome. In this case, there are pairs. So it is called as palindromic sequence. Now this sequence is recognized by this restriction enzyme. Now restriction enzyme, an example for restriction enzyme is EcoR1. This is the restriction enzyme. This is restriction enzyme. Now what EcoR1 does? This enzyme recognizes this sequence. This enzyme recognizes this sequence. So it recognizes the sequence and it sees if this DNA has methyl group attached or not, because that is what we started. If it has methyl group attached, then it is bacterial DNA. If the DNA doesn't have methyl group attached, then this EcoR1 goes and cuts it or cleaves it. This goes and cuts it. Now let's say it has cut it like this, that is ATA, then TC. This much is it has cut. It has cut over here. Now it became two strands over here, correct? There was one which was CTATG before and opposite to it was GATAC. Now because it has cut, there are two strands now. The strand is CTGATA, which is this one, CTGATA. Here there are gaps again, correct? Here there is two gaps. Okay, the other strand is ATAG and opposite here there is TC and here again there is gaps, correct? In the first strand also there is gap and in the second strand also there is gap, and this gap, this region is called as sticky ends. This region is called as sticky ends. Okay, this region is called as sticky ends. Now this is used in order to fulfill, let's say, some enzymes. For example, in some process, let's say there is a human insulin production which has to be done inside the inside. That time, let us say the DNA is taken and then it is having this methyl group, and later on this EcoR1 cuts it into the particular sequence. At that point of a time, this DNA opens, and here outside these two pairs, which one? The ones which are having sticky ends, they are there outside with insulin attached. That is placed inside this. So the bacterial DNA now is having insulin which is attached from outside. So in this restriction enzyme helps because it cuts in the particular sequence every time. It cuts in the particular sequence every time. So in this way there is a recombination which is happening. There are two combinations over here, right? So in this way there are recombination which are happening. Let's say this is one end and this is another end outside, and in between there is insulin which is attached, and then it is joined and it is sent as a whole over here. So it goes inside. So now the bacteria is having insulin within itself. So the production will also increase. In that way, the insulin production will also increase. So this is one of the methods, one of the key tools to accomplish the recombination, and this is a part of the DNA technology. So I hope you have understood about the part 3 sequence in our biotechnology series. At the end of this e-paper, there are a few multiple choice questions for you to answer. You can attempt those. Thank you. I'll see you soon in the next one.